Hi, everybody. Good afternoon.
Hello.
How's
Hello. I
everybody?
just wanted to let you know that Yasmin is having some problems logging in, but she should be here soon.
Thank you, Samantha. Okay. So yes, it seems like, from communication that I'm receiving from students is that some folks are getting sick these days. We have a couple folks sick in our group here in microbiology. So, send some positive thoughts to your classmates because people are getting something right now, whether it's COVID or not remains to be seen. So, okay. Welcome back. I hope you're doing okay. If you do get sick, please reach out to me. I'm always willing to extend assignment due dates and things like that if you need it for illness. That's quite okay. All right. All right. So we're going to continue with chapter 13 conversation here today. Just keeping you up to date. We are now 2 weeks away from our next lecture exam, right? Lecture exam 2 will be on October 21st. At least that's what it's currently scheduled for. At this moment, I think we can make that, but we'll see how things go. Okay. Do you have any questions right now before we continue? Okay, gang, let's see what we can do here. We're going to start with that transcription and translation homework assignment again and see if you've been able to make any progress with that, okay? So I'm going to bring that up into the Word document here. Okay, you recall from last time that we were talking about what it meant to transcribe and translate this sequence of DNA, right? DNA 5' TAC, GGA, ATC, GTC. And we said that the correct answer for the RNA that would be made is 3' here on the left, AUG, CCU, UAG, CAG, 5'. You all did very well with that. Good job. The wheels started to come off the train a little bit when we got to the translation piece of this. So I'm going to ask you again in the chat window, if you will please put in your sequence that you have decoded for the amino acids. What did you get? Hopefully you've had a chance to go back and reconsider this. Okay, so I'm just going to give you a few seconds, so please type in your response. Okay, it's okay, Yasmin. Please put in your response for the translation of the protein, okay? So please put in your amino acids that you think would be made from the RNA transcript. Okay, all right. All right. So how about some more folks? We don't have enough people participating here. More of you need to contribute. Okay, if you don't know, then you can just say that, but please don't leave a blank message here. All right. Thank you, Rachel. I appreciate that. All right. Okay. All right. Okay, so it looks like most of you don't know. And so just go ahead and take a look at the answers that were suggested in the chat window. I appreciate everybody trying. So Chelsea, Yasmin, Elena, it looks like you put some sequences in there. Okay, so let me say that in the chat window here, there are still no correct answers. There are no correct answers yet in the chat window. Okay, so I'm not going to hand this to you, but I'll give you another hint. Okay, I'm not going to hand it to you. I'm going to give you another hint. Okay, So here's my hint coming in the chat window. And my hint for this translation piece is actually coming in the form of a question, okay? Here it comes. What end of the messenger RNA does the ribosome attach to when it begins translating the messenger RNA? That is my hint to you. Okay, that's my hint. And so I'm going to ask you again to go back and hash this out over the weekend, okay? Because this is something that if I just give it to you, it cheapens the lesson, all right? You need to think about this. You need to really think about this. Okay, so please go back and we'll try again next week. All right. Is there anything that you would like to ask? I mean, I'm not going to give you the answer, but if there's something you'd like to ask about this topic, I'll try to give you a subtle hint or a subtle response. Okay, folks, give it the old college try. So Samantha has a comment in the chat window. And so Samantha, yes, 5' N does have something to do with it. Rachel, if you want to answer me, please answer me directly, not to the entire audience. Okay. All right. Okay, folks. So give it the old college try. Really think about it. Okay. Really think about it. That's what I'm hoping that you will do. Okay. And we'll continue with our conversation on viruses. Okay. So we just got chapter 13 started last time. I had told you some fun factoids about viruses, such as, each teaspoon of ocean water has an estimated 50 million virus particles, all that kind of good stuff. What I'm going to start with now is talking to you about the general distinguishing characteristics of viruses. Okay, and let me change my font color here back to black. Okay. All right. So hold on just one moment. I need to respond to a comment in the chat window. Oops. Okay, here we go, folks. General characteristics of viruses. Okay, first off, viruses, we may in fact, have some kind of exception to the rules that science is not aware of yet. But generally speaking, viruses are expected to contain a single type of nucleic acid, either DNA or RNA, but not typically both. Okay, viruses contain a single type of nucleic acid, either DNA or, right? This is an or, it's not an and, or RNA, but not both. Okay, if a virus was ever discovered it had both, then we'd have to rewrite a lot of biology textbooks, which will probably happen someday. Okay. Number two, okay, viruses contain a coat of armor around the nucleic acid, which is called a capsid. That capsid is made of interlocking proteins called capsomer proteins. Okay, so the viral nucleic acid is protected by a protein shell called a capsid. Okay, And, if you have your textbook open at this time, I can give you a couple quick figures to look at if you want to see what a capsid is. You can look at figure 13.2 or figure 13.4, and you can see what is meant by a capsid. Okay. Number 3, viruses, because they are obligate intracellular parasites, the only way they're able to actually replicate themselves, to copy themselves, to reproduce themselves, is to enter a cell and take over the machinery of the cell and force the cell to do its own bidding, force the cell to use its resources to copy and replicate the virus. So viruses can only replicate by taking over the machinery of a cell and forcing it to copy the virus. Okay, it's important that you appreciate that most viruses we are aware of, and there might be a couple oddballs out there that we're starting to be aware of that break some of our rules, but by and large, viruses do not contain their own ribosomes. Viruses do not contain their own ribosomes. So that means that viruses, generally speaking, do not have their own machinery capable of translation. Okay. Viruses also, generally speaking, do not possess the enzymes needed to synthesize ATP. That's something that's typically in the domain of cells, but not viruses. Viruses do not contain their own enzymes to manufacture ATP. Okay. I suspect someday we will find a virus that has some of these things, and that virus will represent a sort of transition zone, between viruses and cells. That probably likely exists. Okay. Number 4, viruses have relatively small genomes, and by that I mean the size of their nucleic acid, DNA or RNA is pretty short, it's pretty petite. But even so, within that small genome, the viruses will contain a limited number of genes that are specific for the virus that support the virus life cycle. Viral genomes tend to be small, but they do contain a limited number of genes to support the virus life cycle. Okay, number 5. Okay, number 5 is this. Viruses typically, and that means there are some exceptions, viruses typically have a very narrow host range. Okay. Okay. And what we mean by that is that typically viruses only infect one type of cell, one species of cell, if you will. It is uncommon for, let's say, a dog virus, a canine virus, to infect a human cell. And it would be odd for a human virus to infect a canine cell, a dog cell. Those are things that are not typically seen. Okay. But because of the way natural selection and evolution take place on our planet, there are exceptions to this. Okay. Okay. And so I'll mention to you, the flu virus. Okay. Take a look at your clinical focus page in your textbook. In my textbook, it's page 367. That's probably not the same page number in your digital text, but if you look close to figure 13.5, you see a special clinical focus page, and it says influenza crossing the species barrier, right? And in there, it will talk about avian influenza, bird flu. It will talk about swine flu. Okay, so we know, because we live in the world we live in, that we do, public health agencies like the World Health Organization, the Center for Disease Control and Prevention in Atlanta, Georgia, they're very interested in monitoring when viruses seem to jump from one species to another, but particularly from an animal species to the humans. That's going to impact our community, and we need to be aware of that. Okay, so consider how influenza virus is able to mutate and replicate in other animals like birds, pigs, there's a horse flu, et cetera, et cetera. Okay, so while viruses do tend to have a narrow host range, and you don't want to build up any anxiety in yourself because you have your dog at your house, and you don't want your dog Lassie to give you a virus. That's pretty unlikely. That's not likely to happen, but we can't say it would never happen. We can't say that. Okay. All right, folks, any thoughts or questions? Okay, so the lab activity that you all have been working on diligently and then submitting today, this afternoon, this morning, was about viruses, the Viruses Explorer. I think that was a really nice lab activity to introduce you to some common viruses that I'm sure you've heard about in some way, shape, or form in your life. Maybe there were some new ones there, like tobacco mosaic virus is probably not terribly common unless you're in agriculture. So it was a good way of introducing to things. Typically, I ask students to take a look at figure 13.1 when we talk about virus sizes. Folks, you can now begin to appreciate virus sizes because you've been doing homework on this for lab and things like that. So talking about viruses that are in the range of nanometers, billionths of a meter, is entirely reasonable, right? So let me just point out a couple of the beasts here on figure 13.1. So of course we have rhinovirus there. Rhinovirus has a diameter of about 30 nanometers. Rhinovirus is the cause of the common cold. We have rabies virus there. It's about 170 nanometers long. Notice the word prion is in there. You remember prion from the category of microorganisms we listed in chapter 1. Prion, of course, is not a virus. It is a misfolded protein. You also have bacteriophage T4, which has a rough length of 225 nanometers. Tobacco mosaic virus. Tobacco mosaic virus has a length of approximately 250 nanometers. And I'm going to throw out a question here. The length you estimated TMV in today's lab activity homework that you submitted, was it close to 250 nanometers? I have a suspicion. Sometimes it may not have been, and we can talk about that later. Okay. A viroid. Okay, viroids are weird little things that I won't spend any time talking to you about in any great length. But if you're looking at figure 13.1, there's this thing called a viroid. A viroid is like a baby virus in the sense that it's got nucleic acid, but it actually does not have a capsid. So a viroid is not a true virus. It's something other than a true virus. And you say, well, where do we find viroids in Mother Nature? Well, a lot of viroids have been discovered in plant biology and actually impact agriculture. Studies have been done at the USDA, the United States Department of Agriculture, where viroids are being intentionally introduced into some of our crops in order to shrink the size of trees. The idea here is that it has been discovered that in some cases you can infect a species of tree that produces fruit. You can infect the tree with the virus. The virus causes the tree to be physically smaller, so it takes up less surface area on land, which means you can plant more trees in the same plot of space on a farm. But the interesting thing is, if you get the right viroid, you make the tree smaller, but it does not reduce the amount of fruit the tree makes. So you can get more trees planted in the same amount of space and potentially double the amount of fruit you get out of that acre of land. Okay, so things like this are being explored by the USDA and other people that are into agricultural biotechnology. Okay, vaccinia viruses there, bacteriophage M13. As you know, every type of cell on the planet gets virus infected. Every type of cell on the planet has fossilized viruses in that. I mean, there's no question about it. And then we have the good old Ebola virus, of which there was a scare a few years back. Remember that, when there was a scare and some Ebola patients were airlifted to the United States, and people were really, really worried, like, oh my God, let's not screw this up, because we don't want to, we don't want an Ebola outbreak in the United States of America. Okay. So let me just ask you one question in the chat window, and then we will move on. Have you read a book called The Hot Zone? Have you ever read this book? Okay, all right. So, folks, this winter time in between classes, or maybe, hey, put it on your summer reading list for summer 2022. Give yourself a period of time when you're not burdened by the realities of life. So maybe you have to wait till retirement, I don't know. But at some moment you feel you have the luxury of time that you can read a book just for kind of enjoyment. The Hot Zone, let me bring it up on Amazon. Okay. It's a book by Richard Preston. There it is. The Hot Zone, the terrifying true story of the origins of the Ebola virus. This is a true story relating to the known origins of the Ebola virus. And it also describes a set of circumstances where there was a lab in Virginia, just down the road from us, not too far, that nearly let the Ebola virus get out into the local community. And boy, there would have been hell if that had actually happened, okay? So it is a true scary story about Ebola in the United States of America. And I encourage you to read it, because certainly after having taken this course, you will appreciate a few more things about the microbiology of viruses. You know, this was published in 2012, so it's almost 10 years old, almost 10 years old. Okay, so it's a really neat, true, and kind of terrifying story. Okay, kind of terrifying what actually happened. Okay, let me talk to you a little bit more about virus structure. So I'm still on roughly the same page in your book as figure 13.1, and I just want to make a few more comments here. Okay, first off, the nucleic acid. Okay, we mentioned previously, just above, that viruses are going to have one type of nucleic acid, DNA or RNA. Okay, so I'll repeat myself. One type of nucleic acid present, but in addition to that, I would ask you to add some detail to this. When we talk about the genome of a virus, you know, it's DNA or RNA, there's some interesting things that happen with viruses that don't happen in cells. Okay, viral genomes can be made of DNA or RNA, and those molecules of DNA or RNA can be either single-stranded or double-stranded. They can be single-stranded or double-stranded. That's very odd, because you've been learning about things in chapter 8 with genetics, right? The reason I teach the virus chapter after the genetics chapter is because now you have a little bit of framework, a background to understand the viruses, because I got to talk about DNA and RNA. Okay, so what I'm saying is, in chapter 8 and for much of biology, when you talk about DNA, it's always presented as a double-stranded molecule, right? Double-stranded molecule. DNA is a helix with two strands linked together by nitrogen bases bonded with hydrogen bonds. Well, viruses don't always agree with that rule, okay? So you can have single-stranded, and I would abbreviate that SS, little SS. You can have single-stranded DNA or single-stranded RNA viruses. That can happen. Now, we know that DNA inside the cell is typically double-stranded. So if you say you have a single-strand genome, that's odd. For RNA, on the other hand, when you think about messenger RNA in a cell going through the translation process, messenger RNA is single-stranded. So to tell you that a virus sometimes has a double-stranded RNA is also equally odd, okay? You can have double-stranded, and I would abbreviate that DS, DNA or double-stranded RNA viruses, okay? And so let me ask you to just take a glance here with me through your own textbook. Please take a look. Examine table 13.2. Let's examine table 13.2. Okay, table 13.2. I just want to be sure you're with me, folks. I'm going to ask you a question in the chat window. What is the heading of table 13.2? What's the title of table 13.2? Okay, thank you, Morgan. Does everybody else see the same thing? Okay, all right, very good. Thanks everybody for contributing and staying focused here. Good job. All right, so table 13.2 is families of viruses that affect humans. First, let me say this. I do not expect you to memorize this table. I mean, the reason we have tables is because you can look things up. The reason we have databases is because you can look things up, right? The internet is a giant database. Everybody Googles the heck out of everything. Nobody memorizes anything anymore. That's good and bad, okay? So you don't have to memorize the information in table 13.2. I don't want you to memorize it. Don't waste your time. But I would like you during your reading of chapter 13 to actually read through it sincerely one time and just take a look at things. But what I want to show you is if you look at the first entry of table 13.2, it says class 1 viruses, and notice it says double-stranded DNA. Double-stranded DNA. Folks, let me ask you, what are the class 2 viruses listed on table 3.2? What are the class 2 viruses? All right, good job, folks. You see it's single-stranded DNA, right? Single-stranded DNA. So you get the idea of what I want you to pay attention to. Class 3 is double-stranded RNA. Class 4 is single-stranded RNA with a positive strand. I'll talk to you about that in just a moment. Okay. And so on and so forth, right? If I go to the next page, I have class 5, single-stranded RNA with a minus strand. Okay. So just pay attention to these sorts of details as you read through the table. Don't try to memorize it, but now you start to know what this is referring to when we talk about double-stranded or single-stranded DNA or RNA viruses. Okay. All right. So that's the first point I want to make. Nucleic acid, one type is present, and it can be single-stranded or double-stranded, okay, depending on the particular virus under consideration, okay? some viruses have a certain strandedness to their nucleic acid. Okay, so viral RNAs, okay, so this is an RNA thing. This is not a DNA thing, okay? All right, viral RNAs can have a characteristic known as strandedness. And here's what this means. Let me give you an example here. Okay. If you have take a look at table 13.2, take a look at class 4. Class 4 says single-stranded RNA plus strand. It's telling you the strandedness of that single strand of nucleic acid. So if you have a plus strand, that is the same thing as saying you have a sense strand, S-E-N-S-E. Okay. Folks, the sense strand of a virus basically means this, that when that virus infects a cell, the RNA that goes into the cytoplasm is of an appropriate structure and sequence that the host cell's ribosomes can immediately attach to it and begin making viral proteins from it. Okay, so if you have a plus strand virus, that means the RNA can be immediately used as a proper template by a host cell ribosome. Okay, this means a host cell ribosome can bind and translate the information in this piece of RNA. Okay, if you have a minus strand virus, such as class 5 on table 13.2, if you have a minus strand virus, that's the same thing as an antisense strand. And for reasons we won't go into right at this moment, this means that when this virus, if you have a minus strand virus infect the cell, that minus strand RNA is not an appropriate template for host cell ribosomes, which means for the virus to get its own proteins expressed, you have to go through some additional gymnastics, which we'll talk to you about later. So simple response here, an antisense strand means the RNA is not a suitable template for a host cell ribosome. Okay. All right. So let me just ask you a very pragmatic question. Let me ask you a couple pragmatic questions. And I'm putting this into the chat window. If you receive a needle stick at the hospital where the needle came from an HIV patient, what should you do? What should you do, folks? Okay. Yeah, that might happen, Rachel. You might want to sue, but that's not going to solve the biological problem. Okay. Okay. Right. Okay. Yes. So take a look at everybody's answers on the chat window. And what I'm fishing for is that you should immediately report that event to your supervisor, right? In other words, you should not hide that that happened, right? Sometimes scary things happen, and for whatever reason, you might want to hide it from your supervisor, or you might want to pretend that it never happened. So you go home that night and kind of ignore it and kind of let it fester, right? You don't want to do that, right? You get a needle stick, you want to report it immediately. And, I say this because we're all human. And when things happen to us, sometimes we act irrationally when we get scared. But if you were to get a needle stick from an HIV patient or a coronavirus patient or whatever it might be, obviously you want to report that immediately. Now, in the case of HIV, if you report it to your supervisor, then what can take place, what should be taking place, is that you will be put on preventative medications, so-called prophylactic medications, to reduce the chances that that needle stick is going to lead to an active infection in you. Makes sense, I hope, right? You want to take some medication to try to lower the risk that that needle stick is going to lead to a bad outcome, such as the development of AIDS. Okay, but here's my question to you. Take the emotion out of it right now. Take the emotion out of it right now. And please respond from a scientific perspective with what we're talking about in our conversation. Would you feel more at risk from a plus strand virus needle stick or a minus strand virus needle stick? Okay. And it looks like just about everybody is answering in the positive, in the plus strand, in the affirmative. And that's exactly what I was hoping you would say, right? Because the plus strand, when it dumps into the cytoplasm of the host cell, it can immediately be used to make viral proteins. So what I'm saying is the infection process is typically more rapid in plus strand viruses than they are in minus strand viruses. Although sometimes the timing is pretty negligible, the reality of it is antisense viruses cannot get the infection started as rapidly because it has to go through other steps to get viral proteins manufactured. Okay? All right, gang. So good for you. Please be safe in the hospital, clinical settings. And, if something bad happens, just report it. Just report it and be safe. Okay. Next, we talked about nucleic acid. Let's go down here. No. Technology. Okay, let's go to number 2. Let's talk about capsids. Okay. Capsids are found in all viruses. Okay, capsids are present in all viruses. Okay, this is like chain mesh armor. This is a protective shell of protein. Surrounding the viral nucleic acid. Okay, the individual proteins of the capsid are called capsomeres. And depending on the kind of virus we're talking about, you could have very simple viruses that have a capsid made of one single type of capsomere that is repeated in an interlocking fashion around the nucleic acid, such as building a Lego robot out of one specific type of Lego brick that you interlock it together again and again and again. Other viruses have more complicated capsid structures, and you can have multiple different types of proteins interacting together, locking together, to build more complicated capsid structures. So some capsids are built with a single repeating capsomere, and others are built from multiple types of repeating capsomeres. Okay, so we're talking here about viral structure, virus structure, nucleic acid. All viruses have nucleic acid. All viruses have capsid. And now we're going to go to something that is only found in some types of viruses. And that thing that I'm talking about is an envelope. Okay, an envelope. An envelope sits outside the capsid. Sits outside the capsid. It is found in some, but not all viruses. And so why this is of particular importance to us as human beings is because enveloped viruses are very common in animal viruses, including human viruses. Okay. And hopefully it came out through the homework and lab activities and readings that you're doing, exactly what this envelope really is. You can take a look at figure 13.3, for example, figure 13.3, to see, an artist's drawing of the envelope. I mean, it's just a cartoon, but gang, the envelope is actually a piece of plasma membrane that the virus acquired when it left a host cell. So when a virus is inside the cell and the virus is reproducing itself and making copies of itself, the brand new viruses, I call them baby viruses because they're young, not because they're immature. These baby viruses, they got to leave the host cell so they can go off and infect other cells. When they leave the host cell, they sometimes rip a piece of plasma membrane of the host cell away from the host cell and wrap themselves in this plasma membrane. Okay. The envelope is actually a piece of plasma membrane that was torn away from a host cell as a new virus was exiting the host cell. Okay. And so let me just ask a question here. Okay, let me just ask a question. All right. Let's see. Morgan Ryder. Morgan, can you unmute your microphone and say hi to us?
Hello.
Hi, Morgan. How are you doing this afternoon?
Pretty good. How are you?
I'm okay, thank you. Hey, Morgan, I have a question for you in regards to this envelope business. So we're saying that the envelope is a piece of the plasma membrane of a host cell. Morgan, would you imagine that having a virus with a piece of plasma membrane from a cell might present a problem for an animal's immune system?
Yes.
Okay, what do you think that problem would be?
I am not sure.
Okay, so let me ask you, let me draw you towards the answer, Morgan. Okay.
Okay.
So Morgan, stay with my analogy here. Morgan, let's pretend for a moment.
Okay.
Let's pretend for a moment that you are a burglar, okay?
Okay.
So Morgan, if you show up at my door wearing a black mask and a hoodie, I might be pretty suspicious of you. Would you say that's a reasonable thing to say?
Yes.
Okay. All right, so Morgan, since you want to be a successful burglar, you want to show up at my front door dressed in a way that does not raise suspicion with me. So Morgan,
Right.
what kind of clothes might you want to wear so that I am not suspicious of you?
Just like everyday normal clothes that I would, I guess, wear on a normal day.
Well, I'm a pretty suspicious guy. Can you give me even a better answer?
Something, I don't know, something you would find unsuspicious, like a dress or something. I don't know.
Okay. You're doing just fine, Morgan. What I'm trying to pull out of you is wearing something that people tend to associate with safety and comfort. So how about you put on a police officer's uniform?
Okay, I see what you're saying.
Okay. So, hey,
Yeah.
thanks, Morgan. I appreciate that. So what I'm
Mm-hmm.
saying is if a virus acquires an envelope from the host cell's plasma membrane, that virus is now coated in the very fabric of the animal body's natural cells. And so it will typically confuse the immune system and make the defense cells of the body think that that virus actually belongs in the cell. So it will slow down the immune response, or in some cases, perhaps even defeat it completely. Okay, so what I'm saying is envelopes tend to be a bad thing from a human health perspective. All right, so let's just make a comment about this here. Envelopes typically confuse the host body's immune system, okay? Because the envelope contains molecules that are naturally / normally found on the surface of cells that actually belong in the body. Okay, so viruses are sneaky little dudes, right? They're taking advantage of what the immune system is going to think is normal, natural, and non-threatening. Okay, any thoughts or questions? Okay, all right, folks. So
I have a question.
yeah.
So then what about the viruses that don't have an envelope?
Well, there's no quick uniform answer on that. You know, viruses are sneaky guys, and they typically, you know, if we were to take a survey of viruses that we know make people sick, you're going to find more of them that have an envelope than don't have an envelope. However, you know, even if you're not an enveloped virus, if you're able to get inside the host cell, just getting into the cytoplasm and away from the exterior surface where defense cells like T cells and B cells could bind to you, kind of camouflages you and, you know, allows you to stay in the body longer. So the presence of the envelope is not an absolute requirement for pathogenicity. However, we do find that typically the most pathogenic viruses do have an envelope.
Got it.
Okay. How about other questions? Okay, folks, let's talk a little bit about growing viruses in the lab. Growing viruses in the lab. Okay, so this is taking me in my textbook to page 370, which puts me somewhere around figure 13.6 and figure 13.7. Figure 13.6 and figure 13.7. Okay, so, okay. It's unfortunate that we're not together in the lab this semester to really kind of talk in a more real world setting about these things. But if I'm completely truthful with you, we don't do a lot with viruses in our microlab curriculum. They're just a bit more involved to handle, not necessarily that they're more dangerous than a bacterium. But at any rate, if you want to understand what a virus is and how it reproduces, well, then you got to be capable of growing it in the lab. And if you think about in terms of manufacturing vaccines for viruses, with the most notable exception of the modern technology that's been used to produce the COVID vaccine, which is a totally different ballgame, most traditional vaccines against viruses involve actually growing, reproducing the virus in some kind of cell in a laboratory manufacturing setting. Okay, so we'll talk a little bit about this, and your textbook gives you a little feel for this. Okay. To understand the characteristics of any virus, you typically have to be able to grow it in a lab setting, okay, in the lab. If you can't grow it in the lab, if you cannot grow it in the lab, then you don't understand anything about it, because you don't even know what that virus needs to reproduce. So if you can't even grow it in the lab, you really have very minimal understanding of what that virus is and is capable of doing. So in this chapter, I'm going to focus on, there are hundreds, if not thousands of different viruses we could talk about. So it quickly becomes overwhelming. I'm going to talk to you in this chapter about two types of viruses. I'm going to talk to you about bacteriophages, and then I'm going to talk to you about HIV, which obviously is an animal virus, more specifically a human pathogen. So for this chapter, we will focus our understanding on bacteriophage, lambda, and human immunodeficiency virus. Okay, so as you know from your homework, and if it didn't come out, if it didn't stay with you, bacteriophages are viruses that infect bacteria, right? So even the lowly bacteria get infected with viruses. Okay, so let's talk about growing bacteriophage in the lab. Okay, and so it is somewhat nice that your textbook does have a nice picture here of figure 13.6. Okay, figure 13.6. Let me highlight that image for you, and let me give you a brief rundown on how you actually grow bacteriophage in the lab. Okay, so let me ask, how about Rachel Spittle? Rachel, can you say hi to us?
Hello.
Hi, Rachel. Hey, if I want to grow a bacteriophage in the lab, what kind of cell do I need to provide that bacteriophage?
You would need, I want to say host cell, but that is just like, I feel too plain.
Okay, so Rachel, let me lead you a little bit. I'm basically asking you, if I want to grow a bacteriophage, should I give that virus a plant cell? Should I give it an animal cell? Should I give it a termite cell? Should I give it a protozoal cell? What kind of cell should I give it?
An animal cell?
Thanks for trying, Rachel. I appreciate it. No, you don't want to give it an animal cell. I want to give it a bacterial cell, because this is a bacterial virus, right? If I give it an animal cell, this virus is going to look at you and say, what is that? I don't want that. I don't like that. I'm not going to go in there. Okay, so maybe let's do this. Remember that bacteriophage are viruses that infect bacteria. They do not infect any other kind of cell. They do not infect human cells. They do not infect dog cells. They do not infect turtle cells. They don't infect other cells. They only infect bacteria. So bacteriophage are viruses, they infect bacteria, and that's all they infect. They don't infect other things. Is it conceivable that some process of evolution would mutate the virus so that it could acquire the ability to infect a yeast cell? Well, it's possible. Given enough evolutionary time, it's possible. But generally speaking, bacteriophage viruses, they only infect bacterial cells. Okay? And so I'm not much of an artist, but if you want to grow bacteriophage in the lab, here's basically what you do. Okay, and I'm going to just draw a little bit here. Okay, so you can also use figure 13.6 as a starting point to think about things. Folks, let me ask you in the chat window. Hold on a sec. Do you know what a Petri dish looks like? Everybody know what that looks like? Okay, I mean, basically take a look at figure 13.8, and that's basically the same kind of idea there, right? So figure 13.6, figure 13.8, that's a Petri dish, okay? So if you want to grow bacteriophage in the lab, what you do is you take a Petri dish, okay, and if my technology will work here, okay, so here you have a Petri dish, okay. Okay, inside the Petri dish, there's a kind of substance called agar, A-G-A-R, okay. So, you know, I'm not gonna draw agar because that's kind of like drawing the air, right? Air is everywhere. It's kind of hard to draw, but it's a Petri dish with agar. Okay, agar, for those that are curious, is made from a seaweed extract. It's made from a seaweed extract, and generally speaking, when you make up the Petri dish agar, you will put some sugar and amino acids and other nutrients in the agar so that something like a bacterial cell could actually grow on the surface of the agar because there's a little bit of nutrients in the agar, okay? So when you think agar, think food for bacteria. It's kind of like making a Jello pop. Okay, you make liquid Jello, you put it in a mold, put it in the freezer, and it freezes solid, and you got a Jello pop. This is an agar pop for bacteria. Okay, and so you take this Petri dish with bacteria, and you take you take this Petri dish with agar, and you take a flask that has a liquid in it, and in that liquid is bacteria. What you'll do is you'll take a sample of the bacteria out of the flask, and you will literally spread it across the surface of the agar plate. Okay, so it's going to look a little bit like this in an idealized manner. So I'm going to spread bacteria across the entire surface of the agar plate so that everything green right now is actually millions and 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 millions of bacteria spread across the surface. Okay, so the green is bacteria. And in fact, if you spread it on thick enough, typically the language that microbiologists use is this will be described as a bacterial lawn, a bacterial lawn. And when I say lawn, I really mean the kind of lawn that you have at your home, where your lawn is made of blades of grass, right? But this lawn is not made of blades of grass. It's actually made of bacterial cells. Okay. So you take the Petri dish with the agar. The agar is a food source for the bacteria. You take bacteria that you grew in another medium in a liquid flask, and you spread that bacteria across the lawn. Question in the chat window. Are you with me? Am I making sense, or is this just like you can't make heads or tails of this? Okay, all right, thank you. All right, so the bacterial lawn is, it would be throughout the entire Petri dish. I'm just too lazy to draw it all out. Okay, I'm just too lazy to fill all of this in really, really dense. Okay, what you're going to do next is, if you want to grow bacteria, you're going to take a tube of virus particles. Okay, so you'd be like, well, where did I get the tube of virus particles? Well, there are ways to do this. Okay, there are ways to do this, and I have to start the conversation someplace. And so let's say that I have a tube of virus particles. A tube of virus, this would be bacteriophage, a tube of bacteriophage virus. You would take that tube of virus, the virus particles are suspended in a liquid. Typically, it's something in science known as a tris buffer. Okay, you put this liquidy solution with virus particles in it, and you take it and you take an eyedropper and you drop a couple drops of that liquid containing virus right onto the surface of the bacterial lawn. Okay, so you drop the virus onto the bacterial lawn. Now, the absolute intimate details of how infection take place, I am going to describe to you, but in a little bit further down the road in our conversation. Right now, what I want you to appreciate is, if you drop the virus on the bacterial lawn and you've given the virus the proper host cell, which we know is bacterial cells because this is a bacterial virus, then the virus will infect the cell. It will take over the machinery of the cell, force the machinery of the cell to reproduce many copies of the virus. Well, how many copies of the virus? It depends on the species of virus we're talking about. It depends on the host cell, but I would say several tens of virus to maybe a few in the low hundreds, depending on what we're dealing with. At any rate, what will happen is the virus goes into the bacterial lawn, viruses start infecting the bacteria in the Petri dish, and when the bacteria reproduce and are made, then the virus wants to leave the host cell that it was birthed in. It's like a newborn baby wanting to leave the hospital. The virus wants to leave the hospital. The baby wants to leave the hospital. The virus wants to leave its host cell. And when it does so, when it breaks through the plasma membrane and cell wall of the host cell, it will kill the host cell. And so what will happen is, hopefully I can simulate this on the screen. What will happen is this, that you will get little, oh, good Lord, that didn't work. No. Okay. I didn't want that. Okay. Let me do it a different way. What I'm trying to show you is the viral plaques of figure 13.6. Take a look at viral plaques on figure 13.6. Okay, what I'm trying to show you is that when the virus kills the cells, there are going to be little clear areas. Guess I need to make that a different color, don't I? It doesn't stick out. There's going to be little clear areas that don't have the original color of the lawn of bacteria. Do you see what I'm talking about in terms of viral plaques? Viral plaques. Folks, viral plaques represent an area where the bacterial lawn has been destroyed. Basically, you have a bacterial cell graveyard everywhere you have a plaque. In the plaque are potentially thousands to millions of newly made virus particles and the carcass of the dead bacterial cell hosts that were destroyed when the viruses left the host cell. So the viral plaques have new virus particles and debris from dead, meaning destroyed, host cells. Okay. Folks, question in the chat window. Are you still with me? Is this making sense? Okay. So, philosophically, hopefully when I describe this, the point I'm trying to make is it's not terribly hard to actually grow virus in the lab. It's not terribly hard to grow bacteria virus in the lab. Now, of course, there are technical details about the composition of the agar, the nutrients that have to go in to feed the bacteria. There are technical things about the liquid that the virus is in to keep the structure of the virus stable. There are technical details that you have to be aware of. But assuming you are aware of these technical details, the basic idea is pretty simple. Put some bacteria on a plate of food, put some virus particles on top of the bacteria. Mother Nature takes its course. The virus infects the host cell, takes over the host cell, causes the host cell to reproduce the virus. The new virus leaves the host cell, and as it does so, it destroys the host cell, leaving the viral plaques. Any thoughts or questions, gang? Okay. Next. Oh, I hate when the software does this. Okay. All right, I will fix that drawing later. Okay, I'll come back and fix that later and get it sent out to you. Okay, so now we have the basic idea of growing viruses in Petri dishes and for bacteriophage. If you have no questions with that, I want to just mention a couple ways that you can grow animal viruses. Okay, so excuse me, number 4 is growing animal viruses. Okay, so first off, there are basically 3 ways this is done. Well, honestly, there are more today with some modern technology, but we'll stick with the 3 basic things that your textbook is telling you. Okay, first off, we have embryonated eggs. Okay, embryonated eggs are figure 13.7. Okay, and the idea is literally you take an embryo in an egg and you use a very fine needle to inject virus into the yolk sac, or sometimes the amniotic fluid itself, and from there Mother Nature will take over. It will infect the cells of the embryo, and basically you'll use the embryo as a viral incubator, inside the egg to grow and reproduce the virus. Okay, that's figure 13.7. Okay, embryonated eggs, even today, 2021, still represent the primary manufacturing technique for making most of our vaccines. Okay, that would not be true for the coronavirus vaccine. Coronavirus vaccine introduced some very new cutting edge technology, and so coronavirus vaccine was not made in embryonated eggs. But let me ask you this in the chat window. Here it comes. Have you ever received a flu vaccine, and were you asked if you were allergic to eggs or egg products? Okay. Well, now you know why, folks. Because traditionally, most of our flu vaccine stock is actually made by growing, reproducing, replicating the influenza virus in embryonated eggs. Embryonated eggs. Many, many years ago, basically a lifetime ago for me, when I was a very young man just getting out of college, I spent a period of time working in Marietta, Pennsylvania. So maybe some of you know where Marietta, Pennsylvania is, at what today is the GlaxoSmithKline building, but back eons ago it was Wyeth Labs. And Wyeth Labs in Marietta actually manufactured flu vaccine decades ago. And so when I was there, literally truckloads, I mean, tractor trailer, truckload after truckload after truckload of eggs, chicken eggs, were brought in from local farms into the Wyeth facilities in Marietta, Pennsylvania. And we would needle stick these eggs to grow and replicate the virus to produce the vaccine. Of course, at that time, I didn't know nearly as much as I know today about it, but it was an interesting process nonetheless. Let me ask you in the chat window. Are you familiar with the Wyeth plant that was in Marietta, Pennsylvania? Okay. All right. So obviously we all live different places, folks. So, some of you, maybe Marietta is not in your region of geography. Okay. But if you ever happen to be over by Marietta or Middletown, Pennsylvania, Bainbridge, that's the general vicinity that I'm talking about. Okay, so embryonated eggs is predominantly used even today, and it has been used for many decades to manufacture many of our important vaccines, including influenza. Okay. Oh man, I'm gonna have to take some time in lab to continue this. Let me just make a couple more comments here. Okay, eggs are very useful for manufacturing vaccines, because at least in the United States of America, since we are a nation that used to pride itself, and maybe somewhat still does pride ourselves in our agricultural, I mean, there's wealth and power and stability in having robust agriculture in a nation. So chicken eggs are simply plentiful. That's one of the reasons it was developed this way in the United States, is because there were lots of farms with lots of chickens with lots of eggs. Okay. Eggs are plentiful in the USA. Okay. Okay, so Yasmin, you're asking if someone is allergic to eggs, they can't get vaccinated. Well, Yasmin, that's a tough call. I mean, it depends on how allergic you are. I mean, the best way to know how allergic you are is to go get, to go get some patch testing at an immunologist office and see how bad your reaction is to egg proteins and things. If you have a mild reaction, you could probably get vaccinated with a product made in eggs, and you would probably be quite okay. Maybe a little discomfort and a little redness on the skin, but you'd be okay. But if your allergic response is very hypersensitive, then no, you could not get vaccinated because it would not be safe for you. Okay. Okay, so it's something you really got to talk to with your doctor about. And honestly, today, in terms of influenza vaccine, there are sometimes vaccines that are made with alternative technologies that it would just be better to get that other version of the vaccine. Okay, folks, I'm out of time here. I'm going to stop. I'm going to pause this recording. Welcome back, everyone. Let's see if we can get refocused here in our conversation. Okay. So before I continue by giving you any more notes, is there any aspect of our current virus conversation that you'd like to ask about? Okay. All right, folks, I'm going to share my screen with you. We'll keep rolling here. Okay, so we've been talking about growing animal viruses in the lab. A reference point in your textbook is figure 13.7, where it talks about using embryonated eggs to replicate viruses. And as we said, embryonated eggs is even today still a very commonly used technology, biotechnology, to grow the viruses needed to manufacture vaccines. Okay. So let's see here. There's a couple nuances to this topic that I want to try to draw out of you. And so let's see how this will go. Okay. So let's see. Chelsea Benzinger. Chelsea, can you say hi to us, please?
Hello.
Hi, Chelsea. How you doing?
Good. How are you?
I'm okay, thanks. Hey, Chelsea, let me ask you a couple questions here on the topic of using embryonated eggs. Okay, so Chelsea, when I was talking to Rachel about what kind of cell I need to give a bacteriophage to grow in, what was the correct answer for that question?
Bacterial cell.
Okay, great. So let's think about it. If I'm going to grow influenza virus, if I'm going to grow influenza virus to protect human beings against the influenza virus, what is the best type of cell to give that virus to grow in?
Well, if you're using it to prevent illness in humans, wouldn't you want to grow it in a human to see how it reacts?
Yes, Chelsea, stay with me. That was a good answer. So the point I'm trying to make here is with using embryonated eggs as a technology. Okay, so embryonated eggs have been used historically to grow things, to develop vaccines for people and animals. And Chelsea, you gave me the answer I was hoping for, that if I want to grow the influenza virus for the purposes of making a vaccine for humans to be protected against this virus, then really I should grow that virus in human cells, not in embryonated chicken egg cells, because that's what this is, right? Almost always it's a chicken egg, embryonated chicken eggs, okay? So I'll be coming back to you, Chelsea, in just a moment. But
Okay.
the point I want to make here is that, as nice as embryonated eggs have been, it hasn't been without problems, okay? So sometimes vaccine production is disrupted because a particular virus that you want to develop a vaccine for just refuses to grow inside a chicken egg. Okay? This, I hope this makes sense to you, because think about it. I'm taking a human, in the case of influenza virus, I'm taking a human influenza virus and saying, hey, please grow and replicate in a chicken egg. Well, typically that human influenza virus is not too happy about being asked to grow inside a chicken egg. In fact, sometimes it won't grow at all. Okay, so that disrupts production, meaning you really couldn't use embryonated chicken eggs all the time to make all the vaccines we want. Okay, okay. Question in the chat window here. Am I making sense so far? Any of that argument you would like me to repeat? Okay, thank you. All right, so my next point is this. Chelsea, let me come back to you. So Chelsea, let me ask you, do you know anybody in your sphere of family, friends, or just acquaintances that often say, I don't get the flu shot because I don't believe in it?
Yes, several.
Okay, fantastic. And you know what? I know people like that too, even in my own family. And the truth of the matter is that quite frequently, from the data reported from the Centers for Disease Control and Prevention, it is frequently true that the influenza vaccine is much less effective than we would want it to be. Okay, so Chelsea, stay with me here. Chelsea, if I'm trying to force the human influenza virus to grow inside a chicken egg so I can make a vaccine to protect humans, do you think that forcing the human virus to grow in a chicken egg might stress the virus out?
Probably yes.
Okay, Chelsea, stay with me. You're doing great. So Chelsea, if I stress the virus out and it is quote unquote forced to grow in the chicken cell, do you think that that virus might in fact mutate in some way so that it is more successful replicating in a chicken egg?
Correct.
Okay, stay with me. If I make a vaccine from a human virus that was forced to grow in a chicken egg by mutating its DNA, when I make that vaccine, does it make sense that the vaccine is going to be really good at protecting against the virus that naturally infects chicken egg cells and doesn't really infect human cells that well at all?
Correct.
You see where I'm headed with this, Chelsea?
Yes.
So what is my punchline? Why am I saying that influenza vaccines are often less effective than we would want them to be?
Because we're producing them in a different species, ultimately.
Yes, that's effectively the punchline. Thank you, Chelsea.
Yep.
Okay, so what I'm saying is, lots of our vaccines, and influenza is probably the one that gets the most notoriety on this. I have lots of friends and family that say, oh, I don't bother getting the flu shot. It seems to be worthless and all this kind of stuff. And, as a biologist, it pains me to hear that. But if I'm being honest, there is some truth to that. And the truth of the matter is that the vaccine is being produced against a mutated version of the virus, and because we're forcing it to grow in a chicken system, rather than the natural version of the vaccine that we're most likely to encounter in the natural world and environment of influenza. What we're saying is we need the vaccine to be produced against the version of the virus that's naturally infecting human tissue. We don't need a vaccine against a mutated version of the virus that really artificially infects chicken cells. They're not the same thing. The vaccine is not a very good match. Okay. So let me just summarize that in your notes here. Forcing a virus like the human influenza virus to reproduce inside chicken cells for the purposes of vaccine production forces the virus to mutate. This means we are manufacturing a vaccine against an abnormal version of the virus that is not the primary threat to humans. Okay. All right. So forgive me for asking folks so many times, but I honestly don't know if I'm reaching you because I can't see your faces. I cannot see you. So is this making sense? Am I making sense with this argument here? Is there any questions you'd like to ask about this? Okay, so please don't come away from this and tell somebody Dr. Allen said I should not get the flu shot because the way it's produced sucks. Please don't say that, okay? Even though there might be some truth to it. Honestly, I still get the flu shot, even though I know of its limitations, because even though it is not a perfect match to the virus that I would really want protection against, it's still certainly very genetically similar. And I'm hoping it will provide enough of an immune system stimulation and cross-reactivity to the natural threat that I'm protected from flu. So I still get it. And one of the reasons I still get it is because even though it's not perfect, I'm hoping it will provide some protection. And truth be told, I'm going to say, well, no, I'm talking over 20 years ago now. Okay, so I'm older than more years than some of you have been alive. I did get sick with the flu, and it knocked me down real hard. I mean, I got really, really sick. And I started to develop pneumonia and everything, comorbidities and things like that. It was a very unpleasant experience. And I promised myself I don't ever want to experience that again. So ever since then, I get the flu shot, even though I do know scientifically it may really not be the best thing out there. I mean, it may not be the best thing that we would want, but it seems to be the best thing we can do with our available technology at the moment. I suspect that's going to change with the new coronavirus manufacturing technology that's been highlighted with this pandemic. I don't know how long it's going to take for that to happen, maybe 5-10 years, but I do suspect that most of our vaccines are going to change in how they're manufactured to a manner that's a bit more of a better match for what we truly need protection against. Okay, so hopefully we can anticipate the vaccine technology is just going to continue to get better, better, and better, hopefully. Thoughts, questions? Okay, all right, so let me wrap this topic up in just a few minutes here. So another way of growing viruses in the lab is on a smaller scale using something called cell culture. And that's actually what you see in figure 13.8. Cell culture is kind of the equivalent to the bacterial petri dish. It's kind of like growing human cells in a petri dish-like environment and then infecting the human cells in the petri dish-like environment with a virus and letting it replicate under that situation, okay? So this is similar to growing bacteria in a petri dish. except we are growing human cells and then infecting those human cells with a virus. Okay. All right. So Samantha Van Broekhoven. Samantha, could we chat a little bit?
Yes.
Okay, Samantha. So Samantha, in the previous conversation we just had with the embryonated chicken eggs, we made the comment that forcing a human pathogen to grow in a chicken cell is really not the best scenario. So with cell culture, I'm telling you that we're now going to take human cells and we're going to use them as the incubator to grow the virus particle. Does that seem, Samantha, at face value to be a better solution?
I think it would be a better solution, but if something were to go wrong, then that human would definitely have like setbacks potentially.
Okay, okay, fair enough. Thanks, Samantha. I appreciate your participation today. So, so with cell culture, it has the advantage of giving the virus the exact correct cell it wants. Okay, but, right, there's always a giant but even this is not perfect. Even this is not perfect. Okay. Okay. So, Odalis, could we chat for just a moment, please?
Yeah,
Hi, Odalis. How are you today?
I'm good. How are you?
I'm okay, thank you. So, Odalis, let me ask you. With this cell culture, we have the potential for the virus to be much happier because we're giving it a human cell. So if I have a virus that naturally infects the liver, I could give this virus liver cells to grow in with the cell culture and it should be happy. For example, right, think about the hepatitis virus. Hepatitis is a virus that is known to infect liver and damage liver and cause, disease. So, Odalis, it makes some sense that the virus should be happier. Are you comfortable with that?
Yeah.
Okay. So, Odalis, let me ask you this. Do you think a virus can tell the difference between a liver that's inside your body at this moment in time or some cells that have been cut out of your liver and put into a petri dish?
No.
Okay, thanks for participating. I appreciate it, Odalis. And Odalis, I want to tell you and everybody that the virus can tell the difference. It can tell the difference. So if you have a liver cell that's part of your liver organ living in your abdomen, and a liver cell that's been surgically removed from that liver organ and put in a petri dish environment, that virus can tell the difference. Okay, so let me just say one thing. When your liver's in your body, isn't your liver being perfused by your bloodstream? And isn't that blood carrying all kinds of molecules, including hormones from other systems of the body? So the liver cells in your body are being bathed in molecules and chemicals that are not naturally present in the cell culture petri dish. Because of that, the virus can actually tell the difference between a cell that's part of a natural organ in your body or a cell that's being artificially grown in the lab in a petri dish environment. And so what I'm telling you is the virus, even though it's in the proper cell, it still may really be unhappy with this proper cell because the proper cell has been removed from its natural context inside your body, and the virus can tell that. Okay, So this cell culture technology is only used in a very limited way in research labs across the nation because it does have some benefit for scientific studies. But when it comes to big, large-scale manufacturing for vaccine production and things like this, unfortunately, this technology is not so good. Okay. So let me continue your notes here. This technology has the advantage of giving the virus the exact correct cell at once, but if the cell is itself out of its natural environment inside the body, the virus can tell. That, long story short, means that the virus will not typically grow very well. Alexis Pearson. Alexis, could we talk just a moment?
Hello,
Hi, Alexis. How you doing today?
I'm good. How are you?
I'm okay, thank you. Hey, Alexis, I got a question for you.
Mm-hmm.
What things, and I'm using the thing as a very generic, I'm using the word thing as a very generic thing to avoid giving you the answer that I want you to give me. What thing inside the cell would change when you remove it from the body, which would enable the virus to detect that there is in fact a difference? What aspect, what thing, what characteristic, what features of the cell would change in a way that the virus could detect?
Would it be something that would cause a mutation, like maybe the DNA?
You know, I really like that answer, Alexis. Nicely done. I'm going to accept that and I'm going to continue off your response. So thanks for participating today. What I'm trying to get at here is that when you take a cell from its natural environment in an organ that's bathed in blood and hormones inside your body, and you take that cell out and slap it in a petri dish in the lab, what I'm driving at is that the genes of the cell will change their expression patterns. The genes will change their expression. You will get different types of proteins turned on from removing the cell from your body, and you'll get other genes that turn off because you remove the cell from the body. So there will be this global rewiring of the genetic architecture because you have changed the location of the cell. You have changed the environment that the cell is in, which results in a rippling of genetic expression changes. When the virus is in there, it's going to know that something is different. Something has happened to alter the gene expression of the cell. The host cell itself is not as happy as it used to be because some of the gene expression is a signature of stress at the biochemical level. And if the host cell is stressed, the virus really doesn't want to use it to reproduce. Okay. So, we're trying to figure out ways to get around this with our science and technology, but this is a hard problem to solve. And so let me go to the final topic of what your textbook introduces you to in terms of growing viruses, and that would be using living animals. Okay, now there's no pictures of this in your textbook, but the writing of your textbook that talks about in living animals is still in the neighborhood of figures 13.6, 13.7. Okay, so the third way of growing viruses in the lab is to use full living animals. Okay, well, here's the deal. Using animals is very expensive. Very expensive, right? Because there's all sort of rules associated with the proper care of an animal. If you're going to be a proper research institution, you need to be ethical about how you treat your animals. That requires resources, that requires money. It's very expensive to do that. Very expensive to use live animals. And we come back to this nugget that we've talked about before. If I want to grow a human virus, a human pathogen, for the purposes of producing a vaccine, the absolute best system to grow that virus in is the actual human body. But let me ask you this in the chat window. Here we go. Would it be ethical to grow HIV in living persons for the purpose of harvesting their tissues to make a vaccine? Please tell me there's only one correct answer here, right? We cannot do that. That is not ethical. It is wrong to sacrifice a human life for the purposes of making a vaccine. So there's my point. If I want a vaccine against HIV, the perfect system to grow the virus in to make the vaccine is actually a human body. That's not ethical. I can't go around and say, hey, George, do you mind if I stick you with an HIV needle so I can grow up some virus? That's not ethical, cannot do it. Okay. You might say to yourself, well, what about people that through their own life choices or bad luck have already acquired the HIV virus? Can't we use that? Well, that's also not really a good idea either, right? We don't want to encourage that. Okay, we don't want to encourage that. So in the case of HIV, we are very limited in growing this virus. Okay. HIV is human immunodeficiency virus, right? It is human immunodeficiency virus. Okay, HIV as a virus will not infect a dog cell. It will not infect a plant cell. You might say to yourself, well, what about FIV, feline immunodeficiency virus? Well, okay, FIV is for cats. It's not for humans. And it's not the same virus as HIV. FIV is a different virus than HIV. FIV is optimized by Mother Nature to infect cats. It is not optimized to infect human cells. HIV is optimized to infect human cells. It is not optimized to infect cat cells. In fact, it doesn't reproduce in cat cells. So what we found out in the research community is basically what you need to grow HIV in a full living animal are chimpanzees, because chimpanzees are genetically similar enough to us that we can get the virus to grow in there. Okay, so very few animals will support HIV replication. Chimpanzees are one animal that does. Okay, so ask yourself again in the chat window, ask yourself this: should we infect 10,000 chimpanzees to make an HIV vaccine? Is that something that you would want to do? Okay, I mean, you know, I would say that is not as questionable of an ethical consideration, but it's still, I mean, I personally don't want to do that. I don't want to do that to an animal. Okay, to me, that's kind of gross. Okay, so the other thing is this: when we want, if we want to study HIV and what it does in terms of causing the development of AIDS, and that's a topic we're going to talk about next lecture. If we want to study this and understand it, then ideally we would use a human, but that's not ethical, so we can't do it. So, well, we could potentially use a small number of chimpanzees to do it, but here's the kicker. While chimpanzees are genetically similar enough to humans to allow HIV virus to grow and reproduce, there's an interesting oddity here. Chimpanzees never develop AIDS. They never develop AIDS from HIV. So if you want to understand AIDS, acquired immunodeficiency syndrome, and you allow the virus to replicate in an animal like chimpanzees, where the animal itself does not ever develop the disease, do you think that's really a good model system to understand AIDS? Not so much, okay? So this is the problem we have in the scientific and research community in trying to understand HIV and its development into AIDS, because we don't have a lot of systems we can reproduce this thing in the lab with in a faithful way, in an ethical way that gives us meaningful information about AIDS. It's a real problem. Yes, we can study people that unfortunately through their own bad luck or perhaps poor life choices have developed AIDS. We can study them and look for commonalities, but that's much less powerful than being able to reproduce it in a controlled setting in the lab. So, long story short, folks, we still do not have a working HIV vaccine. Okay, so chimpanzees will reproduce HIV in their cells, but curiously, chimps never develop AIDS. So chimps are not a very good scientific model for understanding AIDS. Okay. Okay, folks, let me take a quick look here. All right. So, the next time you hear someone getting upset at stupid scientists for not being able to solve problems, please understand that there are some there are both ethical and technological hurdles in answering some of our questions. If we would throw ethics out the window, as maybe you could argue was done with some of the grotesque experiments by the Nazis in World War II, then we could probably make more rapid advances with things. But that would come at a horrific cost to humanity. Okay. All right. Do you have any thoughts or questions at this time? All right. So folks, thanks for your attention on that. I'm going to stop recording because I'm going to stop with lecture at this moment.